This recording is part of a series recording that's covering various cell transport processes. This recording specifically is going to be looking at active transport processes. So we're going to start just a quick review that active transport processes require the expenditure of energy. The substances do not move down a concentration gradient. They go against a concentration gradient. They do not go high to low. They actually can go low to high. And you do not reach equilibrium with active transport. We're going to look at a few types of active transport. So the first one I'm going to look at what we refer to as primary active transport versus secondary active transport. So primary active transport is depicted right here. So this is depicting an example of primary active transport. So with primary active transport, we have a transport protein, which you see right here. Okay, so this here, it's found in that cell membrane. It directly breaks down ATP, and it uses that breakdown of ATP to supply energy to move things against a concentration gradient. This example is showing you the sodium-potassium ATPase, or it's sometimes referred to as the sodium-potassium pump, which is very important in maintaining membrane potentials, and we'll talk about it at a later time. But here you're seeing with this particular transport protein, ATP is directly hydrolyzed by that protein, and what you're seeing here is sodium is moving, or I should distinguish that this is inside the cell. This is the intracellular fluid. Here is the extracellular fluid. And what you'll see is 3 sodium ions are moving out of the cell in exchange for 2 potassium ions moving into the cell. Now I'm mentioning this because the sodium-potassium pump is very important, and so I might as well tell you exactly what this transport protein does. So 3 sodiums move out of the cell in exchange for 2 potassiums. So what we're getting is we're going to have a high concentration of sodium out here and a higher concentration of potassium ions here. So we're not reaching equilibrium. Now this is considered primary active transport because that transport protein is directly breaking down the ATP. Over on the right, this is showing you an example of secondary active transport. So here in this example, you're seeing glucose, which is a monosaccharide, it's sugar, that's being transported into the cell, and it's being transported along with sodium. Now if you look at that transport protein, which you see here going right across that membrane, is energy is not being directly broken down for these substances to move into the cell. But what allows this to happen is that the hydrolysis of ATP by this sodium-potassium pump that you see here creates this gradient where you have a lot of sodium outside the cell, and sodium's going to want to come in. It wants to reach equilibrium, and so if it has this transport protein over here, it says, hey, sodium, you can come on in. It makes it very favorable for sodium to come in, but this protein will only allow that sodium to come in if glucose is along with it. And so glucose is being co-transported with sodium. So this actually is an example of what we refer to as co-transport. And we can actually even refer to what you see here as symport. Or something, if you have a symport protein, substances are moving in the same direction. Over here with this one, this is actually referred to as a antiporter because we're going in opposite directions. So the movement of glucose that you see on the right is an example of secondary active transport. Energy was required to move it across, but the transport protein did not directly break down the ATP. But the breakdown of ATP by the sodium-potassium pump created a gradient, a favorable gradient for sodium to move across, and in the transport protein allows both sodium and glucose to get across, and so glucose is moving into the cell. There's a lot of different transport proteins that allow things like amino acids and bicarbonate and chloride that actually allow them to come into the cell along with sodium. So that sodium potassium ATPase pump is important in transport of a number of other substances. Now, another example of active transport are the various examples that we refer to as vesicular transport. So we have these portions of the cell membrane that form vesicles around something. And we have two types of vesicular transport. We have what we refer to as endocytosis and another one referred to as exocytosis. So the pictures that you see here are all examples of endocytosis. Endo, that prefix, endo, is a word that means inner or within. So here we have a process in which we're going to bring substances into the cell. It requires a formation of a membrane vesicle, and we have three different types. So the first type that you see, actually we'll go in this order here. So the first one that you see here is phagocytosis. So phagocytosis is a process in which the cell engulfs and brings in a solid particle. So you'll notice here this cell membrane is starting to invaginate. It's going to encircle that particle and then bring it into the cell. So that is referred to as phagocytosis. Phago is a prefix that means to eat. So they refer to as cyto means cell, so cell eating. The second picture depicts a process called pinocytosis. Pino is a prefix that means to drink. So literally pinocytosis means cell drinking. With this process, the cell brings in a liquid droplets which may contain dissolved particles. So that has these substances here are dissolved in water, and so it brings in these substances, this liquid that has these dissolved processes. So you see it starts to pinch off here, and then it should have a little molecule in there, and then it goes into the cell. My daughter actually had mentioned how she remembered pinocytosis, and I'm surprised that she came up with it and not me, is she says when she sees pinocytosis, she thinks Pinot Noir, which is a wine, and so you drink wine, so that's how she remembered it. The last example of endocytosis is referred to as receptor-mediated endocytosis. So this one is fairly more specific. So the substances are very specific. They must bind to a receptor. So you see the receptor here. So that receptor is going to be specific for a particular molecule. That substance will bind to it. You see here, it likes to bind to with a star-shaped substance. And then it creates, again, invaginates, and you see this little vesicle, and then you have the substances that would have been engulfed in here. Example of things that are brought in by receptor-mediated endocytosis would be things like VLDL, LDL, so very low density lipoproteins, So these are types of way we can get like cholesterol and things into the cell. So those are some examples. Now the opposite of endocytosis is exocytosis. So exo means outside, or out of. So with this one, this is a means by which we're going to get substances expelled from the cell. So here is something, say for example, those green dots may be, say, something like acetylcholine. And that's an abbreviation for the neurotransmitter acetylcholine. The vesicle that's surrounded by a membrane fuses with the cell membrane, and then these substances are expelled into the extracellular fluid. This is of significance, one, because this is a means by which neurotransmitters are released in the synaptic cleft. When we talk about synaptic signaling, this will come important. This is also a means by which hormones are expelled from the cell into your bloodstream. So you have endocytosis and exocytosis, both of which are referred to as vesicular transport. Energy is required for these processes to take place. Endocytosis brings things in, exocytosis expels things out of the cell. Now, a great example, kind of bringing different types of, talking about different types of transport and looking at the selectivity of the cell membrane is looking at membrane potential. So in next recording, actually we'll use your membrane potential to kind of tie in what we've talked about with all these different transport mechanisms, because it'll become important when we talk about the nervous system and neurophysiology.